Folks. This is Charles Zimmerman from the New York Botanical Garden, broadcasting live to you all through Sococo, which is this fantastic software that we thank WeDigBio for providing for the digitization event for these next few days. I hope all of you have been having a great time so far, all you participants and hosts. So we're fortunate to use this software here to broadcast to you two of our excellent scientists here at the New York Botanical Garden, James Lendemer, who works with lichens, and Dr. Robert Naczi, who works with Cyperaceae and North American plants of all kinds. So without much further ado, James Lendemer is going to begin with his talk. It'll be about 15 minutes, after which we're going to get started directly with Rob's talk. And then at the end, we'll open it up for questions. All of you can use your microphones or use the camera to ask questions, or you can chat in the welcome room box there if everyone who's figured out how to use that already. So I'm gonna get out of here and leave things over to James. So thanks for everybody for coming.
So once again, thanks, Charlie. So my name is James Lendemer, and I'm one of the curators here at the New York Botanical Garden. And I'm going to be talking to you today about some of the uses for herbarium specimens for studying rare species of lichens, sort of as a proxy for other understudied and neglected biodiversity. So just give me a second and I'll put the little PowerPoint up, get that started. Great. So everyone should be able to see this. So before I start and delve into the story that I'm going to tell you about today, I thought it would be helpful to do a little bit of a thought experiment. Now, I can't really see that all of you are going to participate in this, but I trust that you'll do so. So when I say the words threatened native species, close your eyes and just sort of think of it. Let images fill your mind. I figured I'd give you a few seconds of silence. Threatened native species. Okay, that's good enough. So I would guess that the things that sort of came to your mind would be things like turtles or fish, maybe orchids, poppies, Joshua trees, birds, sort of larger animals or more charismatic things like plants and vertebrates. That's what I would argue really would be considered publicized biodiversity. That's the biodiversity that most of the world thinks about, including a lot of scientists, but for sure the majority of the public. But what I suspect most of you didn't think about, unless you happen to be one of the lucky people that gets to study lichens and other cryptogams, is all of the other biodiversity on Earth that makes up much, much more of the life on Earth in total. So that's things like insects, fungi, many types of invertebrates, bryophytes, moths, all of that. And so you can see that sort of on the left. And that's what I think a lot of people, including myself, would argue is the biodiversity that does the bulk of the work to sort of sustain our planet, but at the same time is not what is really publicized. So they don't get a lot of attention, for better or for worse. And so that's what I would call neglected biodiversity. It's neglected. And if you really think about it, I'd like to take a moment and sort of break it down by the numbers. You know, obviously estimates vary, but there are estimates that somewhere along the lines of 12 million species exist on this planet. Let's put a little pie chart up there. Of those, about 88% are undescribed, so they're unknown to science, or they are awaiting description somewhere in a museum collection somewhere. And that leaves 12% that have been described. Of those 12%, or 1.7 million species, if we look at that as a pie chart again, only 18% of the 1.7 million species that have been described to date comprise the species that we would think of as publicized biodiversity, those vertebrates, those sort of showy vascular plants, things like that. That means that leaves 82% of life that has been described to date, hundreds of years of scientific inquiry, neglected. So with that in mind, I'd like to talk to you about one sort of group of neglected biodiversity, and that's lichens. So what are lichens? Lichens are often thought of by many people, especially in the public, as the corals of the land. Now, that's not entirely accurate, but they do form symbioses, just like corals, and they're extremely showy. And just like corals, they form these sort of elaborate and complex communities that sort of serve a home and a host for lots of other forms of life. So they're extremely important. It just so happens that they're also more diverse than corals, and they associate with more symbionts than corals do, or at least more algae than corals do. If there's a coral person in the room, feel free to correct me on that. So lichens are a lifestyle, and they're a lifestyle that's this symbiosis formed between a fungus and an alga. So when these two things get together, they form a lichen, and you can see that in the left there, the picture on the left, that's a lichen. That's old man's beard, Usnea. And if you take a section through that lichen, you'll see that there's algae, that's the green cells in the picture on the right, and then there's fungus. And the fungus sort of encapsulates the algae and provides a place for it to live in exchange essentially for food and nutrition. That's the short of it. If you want more information on lichens, contact me and I'll be happy to discuss it. So it's important to realize that when we're talking about lichens, we are talking about fungi. So if you're talking about a species of lichen, it's a single species of fungus. So in this picture, I've shown you Ramalina stenospora. That's the name of the fungus. The reason that we name lichens based on the fungus rather than on the other components of the symbiosis is that the single species of fungus is the only one that's constant across its entire range. The same species of fungus, in this case Ramalina stenospora, can associate with one or more different algae, depending on where it's growing. So the fungus is the partner that we name the lichen on. And so when we're talking about lichen species, we're really talking about fungi. So the species that I'm going to focus on today and tell you a little bit of a story about is Usnea angulata, also known by the common name fisherman's dream. Usnea angulata is a fruticose lichen, so it's a big bushy one, and it grows in sort of these long tufted colonies that hang down from the branches of trees. You can see a picture here on the left of two of those colonies. Now these things aren't small. They're several feet in size. Sometimes they get to be meters in length. They're quite large and they're quite charismatic. The reason they're called fisherman's dream, or the reason this species is called fisherman's dream, is that if you look up close at the branches there, you can see that they have little ridges on them, little wings where the branches attach. And that sort of is reminiscent of the vertebra of a fish skeleton. So I sort of think that it's what fishermen would be dreaming of at night if they were dreaming of lichens. If you can't see those ridges and you can't see those wings, I've inserted a few arrows there pointing to places where you can see the branches have thinned out a little bit and formed those sort of distinct ridges. It's very, very, very unique. No other species in North America does this. So this is a species that's so conspicuous and beautiful that even non-lichenologists paid attention to it a long time ago and have continued to date. So it was first found in southeastern Pennsylvania in Lancaster County, near the city of Lancaster, by Henry Muhlenberg, who was one of the earliest botanists to work in Eastern North America. And he found this lichen near his home, growing near his home, in about 1803. And he wasn't the only one. Over time, lots of other botanists collecting all over the eastern United States collected this species too. Here's a photograph of a specimen from the herbarium here at the New York Botanical Garden. And you can see, again, you can get a sense that for a standard herbarium sheet, that's a pretty large lichen. And this is a specimen that was collected in northern New Jersey by Co.F. Austin near Gloucester, near the Palisades, across from what is present-day Manhattan. So over the span of time, up until about 1935, botanists collecting, doing their work all over Eastern North America collected this species. And you can see that they found a fair number of occurrences throughout the Appalachian Mountains, the Great Lakes, and also in Texas. This is a pretty standard distribution that we see for a lot of lichens. So this distribution map is actually developed from all of the specimens that we've seen from all of the North American herbaria and European herbaria that would be likely to hold specimens of this species. So not only are these digitized specimen records that I've actually transcribed from digitized sort of labels that were put out there as part of the various NSF-funded projects, but they're also specimens that we physically borrowed on loan from herbaria and confirmed to make sure that the identification was correct. So you can see this is all of these dots represent the entire known range of this species in North America prior to 1970. But if you actually look at the dates of the collections, they stop at 1935. That means that after 1935, there was effectively a 40 to 50 year period where the species was never collected again. Something happened where this charismatic, large, conspicuous species suddenly stopped being collected. So if you actually then look at this distribution, so its historical distribution prior to 1970, you can then compare it to where we know it is now. So this is a summary of all of the occurrences based on specimens that were collected after 1970. And the majority of them were collected by myself and colleagues from here at the New York Botanical Garden and at the University of Colorado in Boulder, and in the University of Minnesota. And you can basically see that all of the populations that are now known are in the southern Appalachian Mountains of North Carolina, Tennessee, and Virginia, in the Ozarks, in Arkansas and Missouri, and in a small cluster around Leech Lake in Wisconsin. Now, what's important to realize is that this distribution, this sudden change in distribution, is a real one. We've done work all throughout Eastern North America, and not just lichenologists, botanists too, and other natural historians, members of the public, amateurs interested in lichens. Lots of people have been out looking, and we know with a high degree of confidence that this is where this species occurs now. The majority of the populations occur in the southern Appalachians, the Ozarks, and in this one spot in Wisconsin. Yes, it does. Yes, Debbie, it means that we have gone back to many of the old locations and it is not there anymore. We know it's disappeared from those places. So this change, the change from pre-1970 to post-1970, that's actually a 65% decline in the extent of occurrence of this species. And that's a more than 27% decline in the area that the species actually occupies in North America. And it's important to realize that those two numbers, while relatively large, are really quite large overestimates of the actual decline in the area that it occupies and the extent of its occurrence, just because of how those numbers are calculated. There's standard ways that that's done for the IUCN. So we know that this species has suffered a significant decline. Now that alone would likely mean that it's endangered or threatened, but it would be one thing if it was still doing fine. Unfortunately, what we've discovered is that all of the majority of the populations in the southern Appalachians and elsewhere occur on conifers. And they occur on conifers in forests that are along rivers and waterways where there's a lot of humidity, and they tend to only occur in old, mature forests. So you can imagine there's not a whole lot of that left in Eastern North America. In the southern Appalachians, where by far the majority of the known populations still exist, oh, sorry, Minnesota, not Wisconsin. Thank you. Leech Lake is in Minnesota. So the majority of the populations that are left are in the southern Appalachians, and almost all of them occur on hemlock. So Tsuga caroliniana and Tsuga canadensis. And that's really important because if you know anything about what's happening to the forests in Eastern North America, and especially with hemlocks, you know that they've been suffering a huge catastrophic decline because of invasive adelgids. So much so that many parts of the southern Appalachians, almost entirely all of the hemlocks have been killed. So here's a view of the Chattooga River in Nantahala National Forest in western North Carolina. You can see sort of there in the center of the photo, there's a dead hemlock. And you can sort of see some green stuff, maybe if you squint your eyes, hanging down from the branches. Here's another picture from the Great Smoky Mountains. This is in a mature hemlock stand, or at least something that was a mature hemlock stand. All of the canopies of these hemlocks were once covered in Usnea angulata. And when I say once, I mean that this was something that was there 2, 3, 4, 5, 6 years ago. Now, as the trees have died and the trees are slowly sort of rotting and falling apart, the canopies are falling down and all of the Usnea angulata is falling to the ground. That means that the little pockets of these things that were left, the little pockets of populations of Usnea angulata that were left, they're on the forest floor and dying. So it's suffering even more of a decline than it did in the past. So it's in decline even further. And that's really significant. And that's something that's really stirred us to take some kind of conservation action. It's important to realize this species is not recognized as endangered or threatened by the federal government, nor I believe by any state, but nonetheless, it most certainly is. And so one thing that we've done, a colleague here at the New York Botanical Garden, Jessica Allen, she has been doing a lot of work transplanting lichens and trying to propagate them. And so when she took this species, some individuals from a population in Western North Carolina, and she has started to propagate them, from the fallen pieces that fall down to the ground that would die anyway. And so she's actually started to propagate them and transplanted them into trees where they will hopefully grow and be able to be used to be established in hemlocks that still survive and potentially on other conifers in the same habitats where the hemlocks have died. So I hope that conveys to you that that's one way that herbarium specimens and digitization have really fundamentally shaped how we understand the past and present distributions of rare species and have actually spurred us to take real action to do something to prevent their declines. With that, I'll thank some of the organizations and people that were involved in this. And rather than take any questions right now, I'm going to hand it back to Charlie, and we'll take questions at the end with a combined Q&A session between Rob and myself. Thanks.
Fantastic. So next, we're going to switch over to Rob. So Rob, you can activate your screen and microphone, and I'll let you go.
Thank you very much, Charlie. Thank you, James, for an inspiring and sobering look at conservation and lichens. And I'm afraid that what I'm going to present is also a sobering story, but it's one that I will leave you with hope, because there really is a reason for a great deal of hope. So at this point, I'm going to share my screen. Okay, what I'm talking about today are intertidal plants, and I'd like to introduce those. These are plants that grow in the zone between extreme low and high tides, and they are a distinctive group of plants because they live in this environment in which tides alternately rise and fall twice a day, and only certain plants are able to tolerate that. You can see the upper limit of high tide where the tree line begins. That's the uplands. And then you can also see extreme low tide, and that's where the water is presently right now in this view of the Hudson River. One example of an intertidal plant is the estuary arrowhead. And in these views, you can see the plant itself in flower. It's quite small, but in aggregate, it's actually a dominant and an indicator of healthy intertidal systems throughout the mid-Atlantic states. Well, why are these plants important? Why would we want to study them? First of all, they're poorly known, and that includes among botanists, even those that study the conservation of mid-Atlantic plants. They're extremely poorly known. Another reason is that what we do know about them lets us know that several of these species are restricted to the intertidal habitat. And in fact, many of them are geographically restricted. So as an example, I show you the estuary bidens, Bidens bidentoides, a member of the sunflower family. What I'm plotting here on this map, based on specimen data, many from digitized specimens, is the total global distribution of this species. And this includes historic as well as current day populations. Many of these are no longer extant. What we see is that this plant occupies only 7 river systems in the entire world. This is an endemic plant of the mid-Atlantic states. In other words, its total world distribution in nature is restricted to the mid-Atlantic states. Another reason for studying intertidal plants is that the habitat itself is quite rare. So for example, many locations are unsuitable because there are no intertidal zones on the river. Instead, the bedrock comes directly down to the river, as we see in this scene. Another reason for studying intertidal plants is that the intertidal habitat is extreme, and extreme habitats select for extreme adaptations. And this is an avenue of research that is completely untapped. but we would expect that a look at intertidal plants would reveal some very interesting and possibly useful applications. Also, intertidal habitats from a conservation standpoint face multiple threats, including such things as climate change, warming, sea level rise. And then another point, and this is the one I hope by now I've convinced you that intertidal plants are important, but I'll end with this one. Intertidal plants provide a range of ecosystem services. And as we think, while we're still in hurricane season, this is especially relevant. So among the things they do is they supply food and shelter for wildlife. They also stabilize shorelines, and the one that I'm showing here is one of the cord grasses that grows very tall. It gets well over 2 meters tall. It and the others stabilize these shorelines, and as a result, they dampen the destructive effects of storm surges. Where we find healthy intertidal systems, we see less damage from severe storms. So the take-home message here is that given their restrictiveness and the numerous environmental threats that they face, intertidal plants are sensitive indicators of environmental health. As we study them, we can learn a lot about the environment and where the deficits are that we can work to restore. So my objective with this study is to conduct conservation assessments of those plant species that are restricted or nearly restricted to intertidal habitats. And what I mean by nearly restricted is a few of these species occur in small populations in a few other environments like lake shores in which the water levels fluctuate dramatically throughout the season. But the majority of the populations are indeed in intertidal zones. Well, most of my studies have been in the Hudson River estuary. And one of the things about the Hudson River that's truly remarkable is its length. The tidal reach of the Hudson goes all the way from the mouth of the Hudson River in New York Harbor, north of Albany, the state capital of New York, over, well, 153 miles presently, but originally it would have been longer because what stops the tidal influence presently is a dam. And this is a river that has a relatively moderate tidal amplitude of 3 to 4 feet. What we did in our work is we first had to document the species in our target group. And through literature review and previous field experience, we found 32 species for our study here in the Mid-Atlantic. These are species that are restricted, or as I say, nearly restricted to intertidal zones. Next, and this is where we get back to specimens, we built a baseline. And we did this by reviewing the historic record through herbarium specimens. We mined the herbaria for information. We looked at 8 different herbaria that house collections of Hudson River intertidal species. And in total, we found about 850 specimens. And we used those by georeferencing them. So here's an example of a real species that a real specimen that we studied and a species. This is the river quillwort, Isoetes riparia. And here's a close-up of the label, and you can read it with me. Isoetes riparia, the scientific name, Engelmann, the author for that name. And then high water line, mouth of creek, just above Peekskill. August 1869, William Leggett, the collector. Legit is the Latin abbreviation for collector. High water line, mouth of creek, just above Peekskill. Well, here we go. This is Peekskill, present day Peekskill in this Google Earth view, obviously. And here's the creek, just north of it. It's still there, and the intertidal habitat is still there. I've been there. I explored this area extensively, but unfortunately, the river quillwort is now gone. So this is leading us into the next part of our study. We followed up after we built the historic baseline from herbarium specimens, and we conducted field work to help learn about the present day conservation status of these plants. So we visited all historic sites and then many additional ones that appeared to host appropriate habitat for intertidal species. And we had to work during the daily narrow window of opportunity, about 2 hours before extreme low tide all the way through 2 hours above extreme low tide. And you can see in this view that our coverage is about the same as the historic coverage. And what I want you to realize is the historic coverage was opportunistic. There were no previous studies that set about doing this kind of work. Instead, botanists went out through the years as they could. The Hudson River is an interesting area, and botanists would pick up intertidal plants along the way. So we covered more sites, but the important thing is, in both cases, we have a thorough coverage of the Hudson River estuary. We accessed our sites by kayak, and we would get out and inspect the areas on foot, and we were very thorough. Most of the areas were small enough that we could explore them completely. And then what we did was we set about analyzing the data, essentially comparing the historic to the current occurrences. And we used GIS, Geographic Information Systems, to help us with this, especially in the mapping and the analysis. Well, these are the results. We have, for conservation purposes, 3 groups, and the first group are those species that are secure. Their future seems to be safe at this point. They have an ample number of populations and coverage throughout the estuary. And on this map, you can see we map the historic specimens, the historic occurrences from these specimens in gray boxes, gray squares, and then the current distributions, the populations we documented through our work, in the red circles. And in both cases, you can see the water hemp is essentially throughout the estuary, and there are a fair number of populations, 34 to be exact, that we know. Well, as an example of an imperiled species, I bring you back to the river quillwort. You can see in the past, the river quillwort was known from several populations and throughout most of the estuary, but presently we know only 3 populations with a total of 129 plants. And then an even more extreme example of how the plants are doing in the estuary is the case of Parker's pipewort. This plant used to be common, used to be found throughout the estuary, but unfortunately it's gone. We visited all of the known historic sites, many additional ones that looked appropriate, because I know this species from some healthy populations in rivers draining the New Jersey pine barrens, but we failed to find it. In fact, its last collection from the Hudson estuary was 1944. And this compares, unfortunately, with the situation in the Delaware River, in which the last known collection was 1927. So this is the summary of our 32 species in our target group. Only 19% of them, 6 species, are okay, they're secure. The vast majority of them are imperiled, 65%, and then unfortunately, 5 of the species, 5 of the 32 have actually disappeared. They're extirpated from the system. So, as I said, I don't want to leave you with all bad news. There are definitely reasons for hope. And the first reason is that we have 27 of the species still with us. This is a critical time in history. If we work for the conservation and restoration of these plants and habitats now, we can make a difference before all of them are gone. Let's hope that day never happens. And then hand in glove with that is recent efforts by the state of New York to restore the Hudson estuary. And I show you here the cover of the Hudson River estuary plan, the restoration plan. So back to the importance of specimens and their digitization. When I began this work, I was focused on the Hudson, and I still am, but I've broadened it to other estuaries in the mid-Atlantic. But when I began this work, I had to go about this by myself because at that time, very, very few specimens were digitized, and it was tremendously time consuming. And then as more and more specimens have come online, my efficiency and my impact have increased because I'm able to mine the data from digitized specimens. So I applaud everyone in the community, and I encourage everyone in the community to continue digitizing and I thank everyone for their efforts. So on that note, thanking people, I have a group of people and institutions to thank, first for funding, then also herbaria and curators for access to the specimens, field assistants, and then assistance with the GIS parts. And I'll leave you with my field assistants, several wonderful people, including Charlie, who went out and spent many hours with me slogging through the mud in these intertidal systems. So with that, I'm going to let Charlie take it away, and then we can move on to our question and answer.
Much. That was fantastic. And on a personal note, I'll say it was quite fun to be out there in the mud, listening to Rob, because he knows so much about the species that are out there. So at this point, I'd like to open it up to questions for everybody. And Rob and James, you can come back and join us. And please feel free to enter questions into the welcome chat or use your microphones if you'd like. Please go ahead and get started. I have a question if no one else has one to get started.
Sure. Go ahead, Charlie.
Rob, you talked about the extreme adaptations of these intertidal plants. Do you want to tell us a little bit more about some of those adaptations that you found?
Sure. Now, admittedly, this was something that I did not spend much time studying, but I did make some observations along the way. And one of the things that I documented was a great deal of morphologic plasticity within a species, depending on its position in the intertidal zone. So you can imagine some plants will be closer to the water and others are higher in the intertidal zone, farther away, yet they're all covered by the tides. And so within that, it was really interesting. In fact, it showed across species that those that were closer to the water tended to be shorter and more compact and produce smaller leaves. Those farther away were taller. But I'll also mention this other one, which I think is the most interesting observation, and that is in the estuary arrowhead that I showed early on in my presentation. I documented in a number of sites something that's not in the literature, and that is as tide waters would rise, those flowers that were open would trap an air bubble around their stamens and stigmas. And this could just be coincidence, it could be happenstance, but I hypothesize it's actually adaptive in that these are insect pollinated plants. Some of the intertidal plants are water pollinated, others are wind pollinated, but this one's insect pollinated. And I think what that bubble might be doing, this is my hypothesis, is that it's adaptive because it buys additional time for the pollen grains to germinate and grow their pollen tubes into the ovules and effect fertilization. Without that air bubble, the pollen grains could be washed away by the inflowing water, the uprising tides. So I'll just mention those couple of things, those observations. They need their hypotheses, they need testing, but I'll leave it at that.
So much.
So another thing that I'll mention, and I'll engage James in this, is when James and I were planning for this, we did not confer. It turns out we gave presentations that were actually pretty similar. So if the message here is things are not going well and we need to do something about it, you've heard it from both of us. But what do you think, James?
What, in terms of whether or not we need to do something about it? Yes, for sure. I would say certainly. But we have, we've done a fair bit of sort of doing our best to push forward conservation for the organisms that myself and my colleagues work on. I mean, I would say that, it's really great to see the kind of presentation that you can give about a group of sort of threatened and clearly critically endangered vascular plants like this. It's really amazing the amount of data there are, but it's also great that it's something that government agencies and even the public recognizes is something that we need to actually mobilize to do something about. Whereas there's a lot of species out there, insects, lichens, other fungi, mosses, all sorts of things that are not really protected at all. And yet we have just as compelling data that indicate that something needs to be done. And the unfortunate thing is that that means that in many cases, we have the knowledge to do something, and we know that we're at that same critical point that you are at with the plants, but we lack in many ways sort of the infrastructure and in some ways the willingness publicly to do it. It's a small number of people who are working on that cause. We do our best to drum up support, though.
Right. Well, James, you just worked toward answering the question that just came in from Debbie about what can an individual do. So I'll jump in and then I'll let you finish. We need help in many areas. One is getting out there and helping with the inventory. Another one is working to get us more resources. As we know, in our country, we have chronic underfunding of science. If we could get more for conservation in natural history, we'd be really in a much better place. We're doing okay, but we could always use more resources. So I'll mention those couple of things. And how do you, if you know, affect change with government and funding? Get involved and talk to your legislators. They're not unapproachable. And if you put on their radar, because it should be there, these kinds of conservation questions of what's going on in our own backyards, it can help if we keep the positive pressure on our legislators to then bring it before their legislatures and make some sort of changes for funding, for example. James, what do you think?
Yeah, I agree. I apologize. The little chat window had closed, so I didn't see the question initially. So, I mean, I really would echo exactly what Rob says, but to me, the most important thing is sort of advocacy for these organisms that are otherwise neglected. That sort of course goes with, write your legislator about, the fact that you care about this and that this is something that you and others should pay attention to and that you believe resources should be devoted to. But at the same time, these organisms, which are charismatic, people know what they are. People recognize them as important, very important to ecosystems and to providing ecosystem services for society ultimately. The most important thing, I think, is really that people sort of become advocates for them locally and sort of take them under their wing to educate others about them. I think a lot of people know what a wildflower is, but there's certainly a lot more education that could be done about what a lichen is. And, it's biodiversity in your own backyard that's conspicuous and tremendously threatened. And so that's, to me, there's just a lot of basic education work that people, sort of across all sections of society, there's a real opportunity there for everyone to sort of get involved and sort of tell your neighbors about it. That's, to me, that's actually one of the most important things that you could do.
I go ahead, Debbie, and then I had one other thing to add, but go ahead.
Oh, so I was going to say, and anybody else wants to, I was trying to make the talking heads not be lonely, so I thought I'd add my talking head, and then you guys can turn on your cameras. It's kind of fun, and then I'll turn mine off when I'm done. But so I recently had the pleasure and opportunity to visit CCBER on the University of California Santa Barbara campus. Has a an ecological restoration project that involves restoring over 300 acres, managing those acres for the UC system. And I know very little about ecological restoration. I'm learning, right? And I was asking, what are some of the things that they do? And I was struck by your map of New York. And there's this river, but the river doesn't know about state boundaries. And the river doesn't care where the water comes from that's going into it or going out of it, right? And so how much of this, when you talk about contact your legislators or contact, is about working with people across boundaries like state boundaries? But also when I was learning about the ecological restoration, one of the key factors I was told was the hydrology, restoring the way the water actually behaves going across the land, like you mentioned the dam, Rob. So I was wondering if that's one of the things CCBER mentioned was at UCSB, by restoring this area that they have that is and you guys know the terms better this than I do, but it mixes ocean and fresh. Periodically, fresh water comes into the system. Periodically, salt water overcomes the berm and comes into the system. So in this system, it's mixed. And one of the problems they had was flooding of their communities. And they're finding that as they restore more of this land to its natural state, it was used as a dumping grounds for a while to take, when people would build a building on campus, they would take the earth they were removing and dump it here in this area and spoil it. So by digging it up and restoring it, they're actually solving a lot of water problems. They're solving community problems that are far beyond, the plant itself, right? That have much more effects than just the whether or not that plant still exists.
That's a wonderful example, Debbie. And it sounds to me like what you're talking about is an estuary too. And I want to bring this back to why most of us are here, and that is WeDigBio and specimen digitization. When we want to set about restoration, one element of that is we want science-based information on what organisms occurred there. Mm-hmm. And natural history collections are the repositories that have the historic data that will answer that question for us. Of course, we hope, we expect, and in most cases it's true, that those collections exist for a given area. And in many of our urban areas, they do, because there's been a long history of collecting. So here in New York, we have some of those same concerns: reduction of flooding, for example, minimizing the damage of storm surges. And we in the community are applying much of this knowledge to rebuild the river and how it flowed, but also to restore the plants that occurred there, these intertidal plants, for example. So it's tremendously important. What can people do is your question. One of the things they can do is get involved in WeDigBio and do more specimen digitization, because what that will do then is it will help the policymakers and those who are doing the restoration to more effectively restore these ecosystems. And it really is a wonderful example of how transformative this work in museums can be for all of society.
Thanks.
Yeah.
Thank you.
So should we address the next question? Or?
Thanks. I didn't even pick up on it. So let's see. Go ahead, James, if you want.
Say a piece on restoration, but yeah. So Libby asked, do you find the sort of the recent data mobilization efforts that these things alert us to the existence of populations of plants, or I guess lichens as well, historical or contemporary, that we weren't otherwise aware of? And Rob, I would imagine, for me, the answer to that is certainly yes. In the case of the Usnea angulata, we actually, I knew that there were historical populations from along the Wisconsin River, but I was unaware that there were extant populations in Minnesota until there was actually a specimen that I saw digitized online. I said, oh, wait a minute, and borrowed the specimens. And at first I just assumed that it had been misidentified, but sure enough, they were all correctly identified. And it's very important, because it shows that there's actually probably a really stable and viable population that exists quite far from all the others. We didn't know that was still there, and we thought it was extinct. So that does happen, yes.
And my answer is a similar one to what you just said, James. The answer is definitely yes, Libby. These digitized collections point out things we did not know. And as an example, I learned some things about the Connecticut River estuary. And as a result, last Saturday, I went out and I made some important discoveries. And what led me there were these specimens that I had seen. So very much so, digitized specimens, their access is easy and convenient. I can see them from my desktop, and I can review many of them very quickly. It takes me a fraction of the time and effort that it does when I do all the legwork myself. And the result is it extends my impact for conservation, which is a wonderful thing.
So I would also just sort of like to, I have another story that I'd like to sort of convey as part of this, which is that, these herbarium specimens also are really helpful because there are things that have been lost. There are species, at least for lichens, that have never been seen since they were first collected. And there's an example of that, which was actually one of the first lichens I worked on way back as a high school student, which, believe it or not, was quite some time ago. And so there's actually, the species was found by Louis David von Schweinitz along the Haw River in North Carolina in the 1830s. And it was subsequently described from a single specimen in 1852, I believe. And then found again in South Carolina at one site in the 1890s. And after that, never found again. It's a very unique thing. It's very distinctive, unmistakable with anything else. People had gone out and made some effort to look for it, but no one found it again. No one knew where the original locality where Schweinitz would have found it around the Haw River was. But we recently, Jessica Allen from the Botanical Garden and myself, we taught an Introduction to Lichenology course in Highlands, North Carolina this summer. And 3 of the students who participated in the course, sort of transcending the generations of people from sort of younger researchers with government agencies to, retired people and undergraduates and graduate students. 3 of those people were very interested in this and just so happened to live in or around where this would have been on the Haw River. And so they actually went out and looked for it, and they had an intimate knowledge of the place. And once they were alerted to the specimens and they looked at the specimens, you could tell what the ecology of the species was from the actual specimen. You could tell it was submerged on rocks, periodically submerged. And there's not very many rocks in these rivers. So they knew where to go look for it. First place they went, they found it. And it was the place where Schweinitz originally collected it. And then they used that as sort of a search image, and they went looking for it again and again in other places. And sure enough, they found 5 more populations. What's probably more important is that they didn't find more than 5 along hundreds of miles of river. But at the same time, all of that is because there was a herbarium specimen that had this information there, and there were people who knew enough about the place and about the people that had been there before that they could connect that herbarium specimen to a real place and time now. So I love that story. I think it's amazing.
That's a wonderful story. Thank you.
Is that story captured somewhere? Did somebody write a blog post or
written up?
Oh, yay. Well, that's very exciting.
You heard it first here. Other questions?
I have one. It's
Okay.
Deb again. And I was wondering, you guys don't be shy here. So this is our opportunity to pick their brains. How do you discover specimens? Where are you going? What do you do to discover that they exist digitally as we do this work on WeDigBio? How do you find them?
Rob, do you
what
want
websites?
Sure,
How do
I'll
you
go
do that?
first. Because the landscape is changing so quickly for the better by having more and more and more digitized resources, when I'm ready to do something new, I just do an internet search, and I first turn up what some likely sources are, but I also begin right away with the WeDigBio portal, since that is the central point of access that I know so well. And then if I want to check specific herbaria that have portals of their own, I can do that. But having the fewest number of places I have to check is what I prefer, because there's only so much time in a day, obviously. But I start out with a general internet search and the WeDigBio portal. That's how I go. How about you, James?
And I basically start with a similar portal with a consortium of North American lichen herbaria. And so that's, again, one of these large project portals. And that's basically where all of the digitized records for almost every lichen herbarium in North America and or in the United States, some from Canada and some from Europe as well. They're all integrated there so that you can search in one place. And that's actually how I find all sorts of things that I never would have thought of before. We've been doing work with moving forward on formal conservation statuses and listing of various lichen species that we know, based on our extensive work throughout North America, are in decline or have already suffered declines and are threatened. or endangered. And so sort of the first step is we go to these portals, we go to other sort of herbarium websites that might not be part of that we know of. And we actually just download all of the data. And then for the specimens that we don't know, we don't have either photographs that we can verify or that we haven't verified ourselves already, we often borrow them and verify them to make sure they're right. But that's the first step to actually confirming that we know where it was and where we think it is now. And then we usually use that to actually go out and do boots on the ground truthing, which I'm sure Rob is also, a huge part of it is you find a specimen either in a herbarium or online, and you go there to see if the thing is still there. Sometimes it is, sometimes it's not.
And that's part of the fun too.
Yeah,
but yeah,
exactly.
Absolutely. The ground truthing is an important complement to the initial research with digitization, and that speeds things along. And
it is, I think, really important that these specimens aren't just dusty museum specimens, right? That this is really a continuing life for them. I mean, you can go back to the same place and find the same species there, and it's amazing. And then to document that in time might not be the same individual, although maybe for some species it would be. I think that's actually fascinating, but it's also it's very interesting and fulfilling as well, and important. Continuity, the continuity of observations and records is very important. There's any other questions?
I have another comment. It sounds like going back and checking these digital records, you've also been able to edit information in herbaria. So you've got these mistakes that you're identifying all the time that otherwise would go unchecked. Being able to find things like that easily from a distance, finding outliers and geographic ranges of things, sounds like a great improvement to the resources as well for other botanists and for the future. Do you agree?
It is. It's very important also because when you're moving forward with conservation action, as part of discovery, someone can come along and say, well, there's this record of it in some other place. They can say that there's records that you were not aware of. And so having the ability to sort of do this extensive search that you never would have been able to do 5 or 10 years ago, maybe even more recently than that, is tremendously important because it really does provide you with sort of the ability to say with a high degree of certainty that the statements you're making are accurate. And it sort of lessens the chance that someone will come along later and discover something that you missed.
Yeah, so what we do with using and then annotating and augmenting the specimen information is we improve the specimen record. We increase the value of these specimens. And as James mentioned, it makes these collections remain relevant and remain alive for our work. So they live on through this work too.
Yep.
Thank you both.
Thank you, Charlie.
Does anyone have any more questions? We might as well push this to the end of the hour if anyone does. Now's your time. Okay.
All right.
Well, thank you both for attending and everybody for joining in. Have a great rest of your afternoon. Thanks a lot.
Thank you.
Thank you.
Bye-bye.